Thank you very much, ladies and gentlemen. This next study looks at a mutation, specific mutation within the HIF1 alpha gene as a potential risk factor for localized prostate cancer and its potential to exaggerate the hypoxic response in clinical specimens of prostate tissue. Ever since its discovery, HIF1 alpha protein expression and treatment outcome has been suggested in a variety of different human malignancies, including prostate cancer. If we assume that hypoxia does indeed promote disease progression, then HIF1 alpha mutations may further amplify the cellular response. HIF1 alpha is a protein that is normally and continuously produced in cellular cytoplasm. However, it's rapidly degraded by the cell, as illustrated in the diagram here. This happens in normal oxygen levels. However, in hypoxic conditions or low oxygen levels, this degradation process is inhibited. HIF1 alpha is stabilized, where it translocates into the nucleus of the cell and it forms a complex. It activates a variety of different target genes and initiates cellular processes that promote survival in low oxygen levels. One particular protein that stimulates is vascular endothelial growth factor, which is highly influenced by HIF1 alpha levels. In the recent literature, single nucleotide polymorphisms or mutations within a specific region of the HIF1 alpha gene have been noted and observed in a variety of different cancers. The specific one illustrated here refers to the C1772T polymorphism, which results in a proline to serine amino acid substitution. In initial studies, it was supposed to occur in higher frequency in androgen-independent patients with prostate cancer. Other in vitro studies have also shown that it can potentially stabilize HIF1 alpha protein in normal oxygen, as well as enhance its activity. However, the in vivo role of such mutations have yet to be established. The aims of our study, first of all, was to correlate the frequency of this specific mutation to men with prostate cancer undergoing radical prostatectomies and compare them to age-matched healthy control subjects. Furthermore, we conducted in vitro cell studies to assess whether a panel of benign and malignant prostate cell lines harbored the CT genotype and if this had any effect on its expression of HIF1 alpha under hypoxic conditions. Lastly, we also assessed patients that harbored the CT genotype to see if they had any influence on HIF1 alpha and VEGF marker expression using immunohistochemistry techniques in benign and malignant prostate tissue. In the first part of our study, we sequenced the peripheral blood of 95 patients undergoing radical prostatectomy and compared these to age-matched healthy controls. We noted that there was over a 4 times increase in the frequency of the CT mutation in our patient group. Furthermore, we also noticed that our patients that harbored the CT genotype also were associated with slightly higher Gleason scores. In our in vitro studies, we took a panel of 5 different cell lines. The 2 on the left are benign, and the 3 on the right are malignant. We exposed these cell lines to varying lengths of hypoxic exposure and assessed HIF1 alpha protein expression using Western blotting techniques. Representative plots are illustrated. We then assessed the genotype of these cell lines, and we noted that the RWE1 cell line, which is a benign prostate cell line, harbored the CT genotype. However, it failed to express HIF1 alpha in normal oxygen, despite having this variant genotype. We then took our patient cohort and we analyzed prostate tissue, staining for HIF1 alpha and VEGF protein. We noted that in the vast majority of our patients, HIF1 alpha was upregulated in tumor sections compared to benign sections. HIF1 alpha is a nuclear stain, and you can clearly see the brown nuclei illustrated in B and C as well as in D in tumor sections. We got similar results for vascular endothelial growth factor staining, where the majority of intense staining was seen in tumor sections compared to benign sections. And again, in photos B and C, you see strong staining within tumor sections, and in D, you see weak staining in benign tissue. We combined the biomarker scores in our patient group, and the vast majority of patients followed an increasing trend of biomarker expression in tumor sections compared to benign sections. In this group of our patients, we also noted that they also tended to have slightly higher PSAs than those that did not follow this trend. Lastly, we correlated our patient group to those that harbored the CT genotype to see if this had any effect on the biomarker expression levels. We noted in our study that there was no significant correlation between harboring the mutation and expression of these biomarkers. In conclusion, certainly these polymorphisms that exist in the HIF1 alpha gene in prostate cancer are complex, as our results suggest. This is the first study that we're aware of where it identifies this specific mutation as a potential risk factor for localized prostate cancer. In our study, we noted that biomarkers HIF1 alpha and VEGF were significantly higher in our tumor sections compared to benign sections. However, our results didn't show any correlation with this mutation and expression of these, either in our cell lines or in prostatectomy specimens. Certainly, these results may merit further investigation of this mutation. Thank you.
In view of the fact that you were getting VEGF expressed in greater amounts, did you look at the vascularity associated with the tumours, and was there any correlation?
No, the reason we included VEGF in this study is to argue that the HIF1 alpha is such a main protein.
Absolutely.
That patients, when you do a prostatectomy, render the gland hypoxic, so you actually might be getting incorrect results on HIF1 alpha expression. So we included vascular growth factor as.
When you showed there were vast amounts of increased vascular endothelial growth factor on your tumour cell sections, as far as I can see.
That's correct.
So one would expect there to be some change in the local vascularity around that area. That's presumably what it's there for. Do you think it's teleological?
We didn't specifically assess vascularity. So.
You don't seem to have talked about the patients who have both alleles mutated, so TT genotype patients. You just seem to refer to CT.
When we
the
sequence,
heterozygotes might have a stronger effect than heterozygotes for this.
that's certainly what's been hypothesized in our patient cohort. There are actually no patients that harbor the heterozygous genotype.
Good, enjoyed that. Two questions. One, could you clarify, you're talking about these activating mutations being somatic or genetically inherited? And secondly, has anyone looked at this mutation, which I presume is genetically inherited in your cohort, and looked at it in other tumor types and shown any correlation with tumor outcome?
Certainly the initial discovery of these mutations initially came out in genetically occurring. There's one study that has shown that it can also occur somatically, but again, we didn't specifically look at that. So.
Other tumor types?
It was initially discovered in renal cell carcinoma, and also there have been some variants also in head and neck cancer as well.